Good afternoon all. Myself is Dr. Madhusmita. I am working as an associate professor in Department of Pharmacology, Faculty of Pharmaceutical Science under Rama University. Today I am going to present a topic about the general principles of preclinical screening models. First of all, we know that what is preclinical screening. Preclinical screening is a crucial part of your drug discovery and development process to establish or to identify a new chemical compound by using different animal models before it enter into the clinical trials. Then it is through this chemical preclinical testings or a screening model. So it determines the potential hazards or establishes safe dosage forms and added to the good laboratory practice to decide if the product is safe to enter the clinical trial or not. Next, before we know that is a pharmacological screening model, so we should have to know about this. What are the drug discovery and development process? Drug discovery is the process through which we can identify one new chemical compounds which are having capability to develop into a potential therapeutic agent which can provide for treatment of different chronic diseases. And development is the process through which there is a optimization of pharmacological activities of a new compound and to create a new safe and effective drug for treatment of different diseases. Then importance of screening models for developing a new compounds. As we know that drug discovery and development is a very long process, time consuming, and it requires a huge amount of money or expensive process. For that, pharmacological screening models we are using to reduce the cost of the research work because through these preclinical studies or screening models we can establish the or to evaluate the safety and efficacy profile of a new drugs before it enter into the clinical trials. So because the clinical trials, through this clinical trials, a huge amount of money is wasted. That's why when we get the data of safety and efficacy data for one new compound, then it is helpful for the research work. Then the importance of preclinical work screening models or pharmacological screening models are essential preclinical techniques through which we are used to identify, evaluate, and optimize the new drug candidate for therapeutic efficacy and safety profile of the new drugs. These models are categorized into two parts, or most of the it is contains three parts. One is in vitro models. Next one is in vivo and in silico. In silico means we are using different apps and through websites, this autodoc websites, we are seeing the binding affinity of the drug compounds to different receptors or proteins. And in vitro methods means through this in vitro methods, the cells which are isolated from the body or some cultured cells or some enzymes, separated enzymes through the cells. So you are seeing the absorption of the new compounds or absorption of the new formulations and the toxicity of the compounds and the mechanism of action of the drugs on molecular levels. Whereas in vivo models, this is the main part of a preclinical studies. In vivo methods, we are using different animal models. Most commonly, we are using rodents or some animals which are genetically and biochemically they are similar to human beings. So, thus, these animals can provide the therapeutic approach or safety and efficacy profile of the drugs before we are testing on human beings. Then, through these preclinical testings, they maintain a bridge or gap between identifying compound and initiating a human trials by testing for specific biological action. Means preclinical testing models or screening models is the gap which fulfill the development or creating a new compound or the human trials. This is the in between these human trials and creating a new compounds. It is the middle part is preclinical testing. So this main aim is to evaluate to identify the safety and efficacy profile of the new drugs. Next, pharmacological screening is a crucial aspect of a drug discovery that plays a pivotal role in identifying a potential drug candidate. This process involves the systemic testing of the compound for their biological activity and therapeutic potentials. Means through in vitro testing, so we can through this preclinical studies by using the in vitro models or cell cultures. different cell lines we are using, and through these cell lines we can see the potential of the drugs or the mechanism of actions in cellular levels or the enzyme levels or protein binding levels. Whereas in vitro models, we can evaluate the safety and efficacy profile of the drugs. Then identification of the drug candidate, pharmacological screening allows the researchers to sift through a vast libraries of a chemical compound to identify those with potential therapeutic value, and these compounds are subsequently developed and optimized to become a new drugs. Before that, and what is the mechanism of action to understand, because with the help of these preclinical testing models, we can understand the mechanism of action of these drugs and also safety profile of the drugs. Next, the method of a pharmacological screening model. So for a different screening models, actually we are using different models for different treating of a different type of disease. For example, so for anti-cancer treatment, anti-cancer drugs, so suppose one researchers develop a huge number of chemical compound, but very few compounds can be able to enter into the clinical trials. Why? Because through these preclinical testing models, we can establish whether the drug is safe for the in cellular level as well as the biochemical levels, and some of only compounds can enter into these human trials. Then through this, with the help of this, for example, like anti-cancer, suppose one anti-cancer drug is developed, and we have to use different models like MTT models and like in-vivo MTT models, and another one is in-vitro case we are using like a cell lines. So cell lines means suppose one chemical compound is developed for oral cancer, we are using OSCC cell lines like oral squamous culture cells we are using. Through this cell culture cells, so we can identify what is the absorption profile of the drug, what is the mechanism of the drugs on cellular molecular levels, as well as the toxicity of the compounds, whether the drug is toxic to the cells or toxic to the other biochemical parameters also, as well as through this, for example, in vivo models, so we are using different animals. For example, if researchers have developed some anti-diabetic molecules or compounds which are showing anti-diabetic profiles, so that's why we are using, first we are using the animals like albino rats we are using. Through these rats, we are first creating the diabetes with the help of different models. For example, alloxan-induced type models, streptozotocin-induced type 2 diabetic models, as well as multiple low dose streptozotocin and high-fat diet-induced type 2 models. These are the different models we are using for creating the or generating the diabetes inside these animals. Then we are using the drugs for it and evaluate whether the drug is safe, and potentially they are used for treating this diabetic disease. Computational models means like in vitro and in vivo models, so we are using computational approaches. So this is computational approaches, so we are using in silico studies. In silico studies means molecular docking studies. We are seeing the through molecular levels or receptor binding affinity levels how the drug is working. Next one is, these are the route map of preclinical studies. For preclinical studies, we are using two methods, in vitro methods and in vivo methods. For in vitro methods, receptor characterizations, receptor binding assay, enzyme inhibition, secondary messenger assays, and cytotoxic activity we are evaluating, whereas in vivo method, you are using both the method, toxicity study and pharmacological screening model. So first, when the drug is developed, first we are seeing the toxic level of the drug through this toxicity study. This toxicity study we are conducting with the help of OECD guidelines. These OECD guidelines, different guidelines are there. For example, for acute toxicity study, oral toxicity 125, 127, 425, 423, 427, different OECD guidelines are there. If it is a normal compound like a topical agents, we are using 402 OECD guidelines. According to the guidelines, we are following the guidelines and we are doing the toxicity study of the chemicals. If once we are confirmed that the drug is not toxic, both for the cellular and biochemical level, then it is entered into the pharmacological screening models. For this pharmacological screening models, we evaluate the efficacy of the drug. For toxicological study, we evaluate the safety of the compound. Next, we are preparing one dose. That dose which doesn't have any toxicity, but it is having potential therapeutic activity. And we confirm that determining the starting dose. That one is dose. We are confirmed that that dose can provide the therapy. It is a potential therapeutic agent which can treat such diseases. And these are the commonly used animals we are using for preclinical screening models. Most of the times we are using rodents. Those rodents which are through genetically recombinant technology, they are prepared and they are similar with us. They are genetically and bioeconomically similar with the human beings, and which can show so potentially they can so potentially the drug can affect the organ levels also. Next, what are the challenges in pharmacological screening models? If it is having some adverse effect, some limitations also there. For limitations like every animals or every biological systems is having different metabolic actions, different biological systems. So that's why some variations are, results are having some variations are showing. And some false data also produced because of some circumstances. For example, while pharmacological screening has made significant process, it is not without its challenges. It is having some good effect, and also some limitations are also there, like a false positive and negative results. It through this preclinical testing models, we can get some false results. So like a screening assay can produce a false positive or false negative results, leading to misidentification of a promising compound or newly developed compound, or the overlooking of a potential drug candidates. Next, predicting in vivo efficacy. While in in vitro screening is valuable, translating results in vivo efficacy remains a complex challenge. For in vitro testing, the chances of false result is very less. But if through in vivo study, according to the circumstances or environmental conditions, so there is a chances of producing negative results or false results. So we are getting. Not all compounds that show promise in vitro are effective in living organisms. In vitro, so some in vitro conditions also can provide a false result. And most of the times, in vitro is the most promising effects they are providing as compared to in vivo models, because in vitro is cell lines only, molecular level we are seeing, but in vivo total living organism we are taking, and these living organisms each other, so they are having some variations. That's why false results production is more common in vivo studies. And complexities of the diseases, many diseases have multifunctional causes and multifactorial causes and complex mechanism. Understanding and targeting such diseases often requires a combination of screening methods and deep understanding of the biology. As we know, drug discovery consists of a huge number of disciplines, like it contains of chemistry, biology, pharmacology, toxicology. Then only one drug discovery will be successful. That's why there is a chances of a complexity of the diseases, and we should have to know the pathophysiology of the diseases, how the disease can induce, and how the disease can be recovered. With the help, if we do not understand the all the things, then it's very difficult to evaluate the safety and efficacy profile of these drugs. Next, rapid technological obsolescence. The rapid pace of technological advancement means that laboratories must continuously invest in updated equipment and training, because day by day the equipment quality is improving. So that's why in laboratories more advanced equipment is, or training persons should be required for getting the good results in screening models. Next, conclusion is what? Pharmacological screening is the engine that drives a drug discovery, enabling the identification of potential drug candidate and the understanding their mechanism of actions. With the help of pharmacological screenings, as it is one of the earliest step of a drug discovery process, that's why with the help of a screening models, one drug can able to be developed. Potentially, they can be able to develop into a therapeutic agent, and which can improve or which can create a safe and effective drugs for the development of a human life or a human conditions. means the patient conditions for improvement of the patient conditions. Screening model is a very crucial part of the drug discovery process.